Hi, it's Melissa Etter again, and this is the video on Chapter 2 for Lifespan Psychology. I gave some additional information about these videos at the start of the video for Chapter 1, so if you haven't yet watched that one, you might want to stop and start with it instead. The human body is composed of cells. Within each cell is a nucleus that contains 23 matching pairs of rod-shaped structures called chromosomes. Each chromosome holds the basic units of heredity known as genes. Genes are composed of stretches of DNA, a complex molecule shaped like a twisted ladder or staircase. Genes are the blueprint for creating all of the traits that organisms carry. The set of instructions to construct a living organism is referred to as the genome. Although all humans share the same basic genome, every person has a slightly different code, making them genetically distinct from other humans. Most cells in the human body reproduce through mitosis, in which DNA replicates itself, permitting the duplication of chromosomes and ultimately the formation of new body cells with identical genetic material. Gametes, or the sex cells, ova and sperm, reproduce by meiosis, in which the chromosomes cross over as they replicate, creating two cells that each have 46 chromosomes. They then divide a second time, creating gametes with 23 single, unpaired chromosomes. Gametes have only one chromosome from each pair. Sperm and ovum join to produce a fertilized egg called a zygote with 46 chromosomes, forming 23 pairs with half from the biological mother and half from the biological father. The sperm determines what the child's biological sex will be. Mothers can only contribute an X chromosome via the ovum, while fathers can contribute either another X chromosome or a Y chromosome. An XX will result in a biological female, an XY in a biological male. Twins are siblings who share the same womb. Most are dizygotic or fraternal twins who arise when a woman releases more than one ovum. Two ova are fertilized by two sperm. Genetically, fraternal twins are no different from other siblings, sharing one half of their genes. Like other siblings, most differ in appearance, such as hair color, eye color, and height, but some are very similar. Unlike dizygotic twins, monozygotic or identical twins originate from the same zygote. Identical twins arise when the zygote splits into two distinct, separate, yet identical zygotes that give rise to two births. Identical twins share the same genotype with identical instructions for all physical and psychological characteristics. They share phenotypes that are carried by genes. Some genes are passed through dominant recessive inheritance. In this, heterozygous alleles are paired. Heterozygous means that they are different, while homozygous means they are the same. Alleles are one of two gene variants, either dominant or recessive. If one allele is dominant, that trait will always be expressed, regardless of the allele it is paired with. As an example, the allele for blonde hair is recessive. If it is paired with an allele for dark hair, which is dominant, the person will have dark hair. If an allele is recessive, it will only be expressed if paired with another recessive allele. Blonde hair, for example, requires homozygous alleles, two recessive alleles for blonde hair. When a person is heterozygous for a particular trait, as we see with dominant recessive expression, the dominant gene is expressed and the person remains a carrier of the recessive gene, meaning they can pass it on to their children. In most cases, dominant recessive inheritance is an oversimplified explanation for patterns of genetic inheritance. Incomplete dominance is a genetic inheritance pattern in which both genes influence the characteristic. Even though one allele is stronger than the other, it does not dominate it. An example of this would be the AB blood type, where neither allele overpowers the other, but they combine in their expression. In addition, most traits are a function of the interaction of many genes, known as polygenic inheritance. Researchers cannot trace most characteristics to only one or two genes. Instead, polygenic traits are the result of interactions among many genes. Examples of traits that are a result of polygenic expression include height, intelligence, temperament, and susceptibility to certain forms of cancer. Expression of genes is also influenced by the environment, something known as epigenetics, which will be covered in more detail later in this chapter. Some traits are determined by a process known as genomic imprinting. Genomic imprinting refers to the instance in which the expression of a gene is determined by whether it is inherited from the mother or the father. PKU is a recessive disorder that only occurs when both parents carry the recessive allele. Individuals with PKU cannot produce an enzyme that breaks down phenylalanine, an amino acid. The phenylalanine builds up quickly to toxic levels that damage the central nervous system. Avoiding sources of phenylalanine through diet can prevent some of the deficits. On the other hand, disorders carried by dominant alleles, such as Huntington's disease, are expressed when the individual has just a single allele. Unlike most dominant disorders, Huntington's disease emerges later in life, at age 35 or later. Dominant inheritance of disorders typically result in death before the individual can reproduce and pass the disorder on to children. This table, which is also in your book, Table 2.3, summarizes diseases that are inherited through the dominant recessive inheritance pattern. Some recessive genetic disorders, like the gene for red-green color blindness, are carried on the X chromosome. These are known as X-linked disorders. Males are most likely to be affected by most of the X-linked genetic disorders, such as hemophilia, because they only have one X chromosome. Females have two X chromosomes, so a recessive gene located on one X chromosome will be masked by a dominant gene on the other X chromosome. Fragile X syndrome is an example of a dominant recessive disorder carried on the X chromosome. Because the gene is dominant, it must appear only on one X chromosome to be displayed. That means that Fragile X syndrome occurs in both males and females. This table, which is also in your book, Table 2.4, summarizes some X-linked disorders. Chromosome disorders occur when there are errors during cell reproduction, either during meiosis or mitosis, or due to damage caused after the cell reproduction. The most widely known chromosome disorder is trisomy 21, known as Down syndrome. Down syndrome occurs when 3 chromosomes rather than 2 appear in place of the 21st pair of chromosomes. Although individuals with Down syndrome vary in the severity of symptoms, Down syndrome is associated with marked physical, health, and cognitive attributes. People with Down syndrome experience physical and motor developmental delays, immune system deficiencies that often lead to infection, commonly of the respiratory tract, and wide-ranging intellectual abilities. Down syndrome is the most common genetic cause of intellectual disability. If supported and cared for, people with Down syndrome can experience positive outcomes, but the disorder is associated with early Alzheimer's and other signs of early cognitive decline and aging. This figure, 2.6 from our book, illustrates some of the physical characteristics of a child with Down syndrome. Some chromosomal abnormalities occur on the sex chromosome, or the 23rd chromosome. Klinefelter syndrome is one of the most common sex chromosome abnormalities that occurs in males born with an extra X chromosome, XXY. Symptoms range in severity. Some experience some impairment, such as a high voice, feminine body type, a tendency to be overweight, and memory impairments, while others are unaware of the disorder until they are tested for infertility. A second type of sex chromosome abnormality experienced by men is XYY syndrome, also known as Jacobs syndrome, a condition that causes men to produce high levels of testosterone. As teens, some experience severe acne and poor coordination, but most men are unaware that they suffer from this condition. Females are susceptible to a different set of sex chromosome abnormalities. Some females are born with 3 X chromosomes, known as triple X syndrome. They present physically within the norm, so little is known about this disorder beyond that it seems to be accompanied with low ranges of intelligence and some learning difficulties. Turner syndrome occurs when a female is born with only 1 X chromosome. Women with this disorder are often shorter than average, their ovaries do not develop normally so they do not ovulate, and they can experience a spontaneous onset of puberty, often at an early age called precocious puberty, instead of a gradual beginning. Regular injections of human growth hormones can result in some breast development and menstruation. Mutations are sudden changes in abnormalities of one gene or many genes. They can occur spontaneously or as a result of exposure to environmental toxins. Most of these mutations are fatal. The developing organism often dies soon after conception, typically before the woman knows she is pregnant. Maybe as many as half of all conceptions terminate due to mutations. But sometimes mutations are beneficial. As one example, sickle cells result in a resistance to malarial infection, which is helpful in areas where malaria is common. In fact, it originated in those areas of the world as an adaptation that helped people survive malaria. However, sickle cell anemia can cause significant problems for people living in other parts of the world. Genetic counseling is a medical specialty that helps prospective parents determine the likelihood that their children will inherit genetic defects and chromosomal abnormalities. It involves interviewing and testing the parents for genetic disorders to allow them to make informed decisions regarding biological children. Reproductive technology methods have improved dramatically over the last few decades and include artificial insemination and in vitro fertilization. Parents can also turn to surrogacy, in which a surrogate is impregnated and carries a fetus to term, agreeing to turn the baby over to a woman, man, or couple who will raise the child. Adoption is another option for prospective parents. Adoptive parents tend to have higher levels of education and income. They often spend more time with their children and have more resources. However, adopted children tend to be less successful in school up through college age. Sometimes psychological and adjustment problems persist to adulthood. How the adopted children fared prior to the adoption impacts their ability to adjust, but most are resilient and able to overcome past trauma and neglect if they are able to establish a healthy bond with their adoptive parents. Prenatal testing is recommended when genetic counseling has determined a risk for genetic abnormalities, such as when the woman is older than 35 or when both parents are members of an ethnicity at risk for particular genetic disorders. It is also recommended when fetal development appears abnormal. The most widespread and routine diagnostic procedure is ultrasound, in which high-frequency sound waves directed at the mother's abdomen provide clear images of the womb, represented on a video monitor. A fetal MRI is sometimes done after an ultrasound to provide more detail. Amniocentesis is a prenatal diagnostic procedure in which a small sample of the amniotic fluid that surrounds the fetus is extracted from the mother's uterus in order to conduct analyses to detect genetic and chromosomal anomalies and defects. Chorionic villus sampling, or CVS, can be conducted earlier than amniocentesis, between 9 and 12 weeks of pregnancy. CVS requires studying a small amount of tissue from the chorion, part of the membrane surrounding the fetus, for the presence of chromosomal abnormalities. All of these prenatal testing methods involve varying degrees of risk to the developing fetus, but non-invasive prenatal testing, NIPT, involves only a blood draw from the mother and presents no risk to the developing fetus. Today, it is less effective than the other methods, but someday might be as effective as amniocentesis and CVS. Advances in genetics and in medicine have led to new therapies to reduce the effects of many genetic abnormalities, such as administering drugs to the fetus and conducting fetal surgery. One day, we may be able to treat many heritable disorders through genetic engineering. Here's Table 2.6 from the book, giving more detail on these methods of prenatal diagnosis. Fetoscopy can evaluate the developing fetus by way of a small camera inserted through the amniotic sac. The risks of this procedure include infection, rupture of the amniotic sac, premature labor, and even fetal death. Other prenatal treatments involve using needles to deliver medications, hormones, or transfusions to the fetus. To repair some defects, such as with the heart, lungs, or urinary tract, surgeries can be performed on a fetus. Someday physicians might be able to remove harmful genes, replacing them with healthy ones, and correcting many heritable disorders even before the child is born. Behavioral genetics is the field of study that examines how genes and experience combine to influence the diversity of human traits, abilities, and behaviors. Even traits with a strong genetic component are modified by environmental influences. Heritability refers to the extent to which variation among people on a given characteristic is due to genetic differences. Heritability research examines the contributions of the genotype, or the actual genetic makeup, in determining phenotypes, or the traits that are expressed and shown, but also provides information on the role of experience. In selective breeding studies, behavior geneticists deliberately modify the genetic makeup of animals to examine the influence of heredity on attributes and behavior. Family studies, such as twin and adoption studies, compare people who live together and share varying degrees of relatedness. Twin designs compare identical and fraternal twins to estimate how much of a trait or behavior is attributable to genes. Adoption studies compare the degree of similarity between adopted children and their biological parents, whose genes they share, and their adoptive parents, with whom they share no genes but an environment. Individual personal characteristics are impacted by the influence of multiple genes as well as by the environment. Identical twins consistently have more highly correlated intellectual scores than do fraternal twins, demonstrating some genetic tie to intelligence. Genes contribute to growth, body weight, and body height, but environmental influences also play a role. Even highly heritable traits like height can be impacted by environmental factors such as poor nutrition. Genetics contributes to many traits such as intellectual ability, sociability, anxiety, agreeableness, activity level, obesity, and susceptibility to various illnesses, yet even identical twins who share 100% of their genes are not 100% alike. Reaction range refers to the idea that there is a wide range of potential expressions of a genetic trait, depending on environmental opportunities and constraints. Some traits instead illustrate canalization and require extreme changes in the environment to alter their course. Canalization means that the genes narrow development to only one or two possible outcomes. Canalized traits are biologically programmed. Barring health problems, around the world, infants walk sometime around 12 months, and only extreme deprivation or neglect can impede that development. Gene-environment correlations illustrate how traits often are supported by both our genes and environment. Genes give rise to behaviors, and those behaviors are associated with the environment. Passive gene-environment correlations occur when environments are created for us. Parents are genetically similar to their children. The homes that they create are in line with their own interests and preferences, which also correspond with and support the child's genotype. If parents and their children love music, their homes will be filled with music. In a reactive or evocative gene-environment correlation, a child's genetic traits, such as personality characteristics, influence the social and physical environment, which shape development in ways that support the genetic trait. A happy baby gets positive attention, which makes the baby even happier. An active gene-environment correlation occurs when the child actively creates experiences and environments that correspond to and influence their genetic predisposition, called niche picking. That musical child will find musical friends and enroll in musical classes. The Epigenetic Framework is a model for understanding the dynamic, ongoing interactions between heredity and environment. Genes influence development and experience, yet gene expression is also influenced by development and experience. Not all genes are automatically turned on or expressed. The epigenome is a molecule that stretches along DNA and provides instructions to genes, telling them whether to turn on or off. Throughout life, the environment can also tell genes whether to turn on or off. An illness or trauma can cause a gene to switch on, for example. What you do today can be passed along to future generations. Fathers who stop consuming alcohol 6 months prior to conception reduce the rate of their future children's heart risk. Okay, that's it for Chapter 2, which also means this is the last of the 2 assigned VidGrid videos. If you're taking this class with me as a hybrid in-person course, the first time we meet together in class, I'll actually start lecturing on Chapter 3. If you're taking this with me as an online-only course, these are the only 2 videos I'm going to make lecturing on our textbook. I look forward to meeting all of you soon, at least virtually.